Hello. Yeah. Okay. Welcome to the last afternoon session, and we're going to hear from Riley Doyle on reprogramming the genome. Thanks, Riley.
Thank you, and it's great to be here. This is my first PyCon. My background is actually in biochemistry, so if you had told me 5 years ago that I'd be speaking at PyCon, I would have thought you were crazy. So it's going to be, it's a fun time though, because we're going to talk about something a little different, which is the amazing work that's been going on recently in cell and gene therapy with CRISPR genome editing in particular, and how we have been using Python actually to facilitate this process and make it safer and more effective for patients. So the idea that we work on at Desktop Genetics is to go and use machine learning to make CRISPR genome editing safer and more effective. We're a startup, 5 years old. We're based in London. And it's a pretty interdisciplinary team, combining people everywhere from, say, data visualization and UI design into data science, lots of back-end Python programming, as well as the life sciences as well. I did this work in collaboration with our data scientist, Mark, who's here in the audience. So don't ask me any terribly complicated questions about machine learning, or I'll have to bounce you over that way. And I think this is an interesting example of a theme that we've seen at a couple talks, though, where it's really an interdisciplinary collaboration between people coming from both a domain background, say in this case biochemistry and genome editing, as well as computer science, and the sort of amazing things that we can achieve when we work together, and then Python really acting as a bridge for that. If you want to get a copy of the slides, just send an email, pycon@desgen.com, and we'll auto-reply with this PDF and all sorts of other goodies, which will be handy. By way of an agenda, we'll give you a brief intro to CRISPR, talk about then how we apply machine learning to it, our own CRISPR design process, and some of the future work going forward. So this CRISPR thing, it's a bit of a buzzword. I don't know if you all read XKCD. They did a comic on it actually pretty recently. So CRISPR and the Vim bindings are going to be a big thing. I think I've actually started working on some of those already. So if you're interested, let me know. And I'm gonna make a really weak metaphor here about trying to maybe explain some of the biotech vocabulary in terms of programming terms. So if your cell is a computer, the DNA would be like your Python source files. The genome is just simply the set of all of them. It's all of the source code that's running on this particular machine. The RNAs then would be the processed binaries that are actually getting executed, and the proteins would be the actual objects loaded into memory. So those are things in volatile storage. And CRISPR then is basically said. It is find and replace, let's grep for something, let's edit it out. And really all we're doing then is sort of taking these CRISPR packets and chucking them at a computer and randomly editing its source file in as specific a way as possible to try and affect some change. And in this context, you see there's some really interesting quirks that happen because the genomes and cells, they don't have the concept of namespaces. So everything is sort of all mixed together. And this gives rise to one of the really interesting challenges, which is that you have to come up with a search query, a string effectively, that will locate the region of the genome you want to edit, but not edit anywhere else. And that gets a bit tricky. There's lots of hype about CRISPR. It is actually a big deal. It's one of these things that really overhauls the way we do work in the lab, once in a generation. And so I think it's going to be a really exciting time to be involved in the field. Just how this actually plays out in practice, what would this look like one day in the clinic is typically you have a patient, you're going to extract some cells from them, you're going to sequence those cells and characterize them, and then you're going to go and design a genome editing vector, a CRISPR, and you're going to introduce that to those cells to affect some kind of change. Then you're going to test to make sure there weren't any side effects or unintended consequences of that edit, which is a bit of a trick. And then you're going to say, yep, everything looks good, and then we're going to reintroduce them. And this is a pretty common technique right now in something called cell therapy, used to treat blood cancers as well as to treat rare genetic disorders. Now, one of the other quirks here is that I'm sad to say that all of you are mutants. Everyone in this room has 4 to 5 million mutations in their genome that separate them from the person sitting next to them or behind them, and that you have to take these into account when you're designing your CRISPR sequence. So the mutations not only determine why your DNA is going to compile into something different than the person sitting next to you, but also if you're trying to edit that DNA, you have to understand that the sequence is different as well and take that into consideration. So these are actually the genomes of my 2 co-founders, Eddie and Victor. They're both mutants, but they're not the same one. So I don't know who got the superpowers or not, but we'll see. In terms of the overall process, what happens is, as I said, there's some sequencing. We come in now at the design phase. You manufacture these. This really all gets automated very early on by the magical lab robots, and then you go on and sequence and analyze your data at the end. I just put a couple pictures of the Illumina flow cells that are used to do this, the oligosynthesizers, and then ultimately the cell culture robots, just to give you a sense of the sort of all the high-tech kit that gets involved in this process. There is some really interesting work actually around using Python to automate this and to chain all of these really expensive instruments together, and I'm going to point you to some resources at the end of the deck that'll tell you a little bit more about them. So now I'm going to have a brief video interlude to give you a real overview of what this CRISPR process is all about. So here we have our CRISPR guide RNA and nuclease that actually is going to do the cutting. And you're going to see here the 20-character string that we're going to use to search the genome. This is a guide RNA sequence. And that has to enter the cell and its nucleus, and it's going to go along looking for a place where the sequence of the guide RNA matches to the sequence of the genome. And it's a process called strand invasion, where they will go and they form this complex called an R loop. And then, having found a successful match, the DNA is cut. That's it. It goes in and it cuts the DNA. This triggers a repair process then that does the rest of it. So you see here that it's going to sort of chew back and then extend the DNA to try and fix this double-strand break. And we can actually direct this process by introducing a template sequence to direct the repair down a particular pathway. And this way we can introduce a very specific new mutation to the cell. So this is the sort of replace of our find and replace operation. If you don't do this last part, you just get a deletion. So there you have it. That is how it works in a nutshell. So here are the backup versions, just in case the video didn't play, but we're all good. Again, here's just the diagram. So it's effectively a string search problem. And what we can use this for is a whole bunch of really cool stuff. We can use this to go and create custom models of a particular cancer. We can use this to humanize, say, the heart valves in pigs for tissue transplant. They've done some really amazing work curing both leukemias as well as HIV in patients and in animal models by going in and being able to excise the virus from the genome or to reprogram the immune system to more effectively fight the cancer. And so there's going to be some really exciting work coming out in the next couple of years. And if you want to learn more about that, you can go to clinicaltrials.gov and see the thousands of new projects that are currently under investigation. So where does the Python come in here? Well, there's 2 sort of computational problems we're trying to solve. The first is one around activity. How do we design a guide RNA, a CRISPR, that's going to work? And the second is, how do we make sure it doesn't have side effects? So these are effectively a form of prediction or ranking that we want to look at. So given the guide RNAs in this gene, which one is going to actually cut the best, and which one is going to be the most specific and not cut somewhere else? There are a couple other ways you can also look at this in terms of lower-level biochemical properties or really high-level things like ultimate outcome for the patient. But at the end of the day, it's really some kind of prediction problem based on a large number of variables, and so this becomes a really good application for a machine learning approach. We also found some really interesting bits when we looked at our users' behavior. Even expert scientists are actually really bad at designing CRISPR relative to a machine. They get tired. They inconsistently apply whatever the latest paper was that they read, and they give it much more weight than, say, the larger body of evidence that they've seen so far. They don't really consider anything more than the reference genome on the whole. They don't actually consider the sort of unique genome of the cell they're trying to edit. And the approach tends to be one more of designing, say, the minimum number of winning guides to get an experiment done, rather than designing in a more neutral way, a set of guide RNAs to learn something about the system, right? Say that we're trying to come up with a training data set. Really the machine excels at making sure that it's sufficiently diverse and chosen randomly and follows sort of good experimental design practices. So what we did is came up with a set of predictors, our features, and those were based both on the biochemistry as well as the sequence itself. So this is sort of a laundry list of the various biochemical features we've looked at. I can go into these in more detail if you're interested, but the short version is we just we read a lot of papers and pulled these from the literature. We also went and looked at the sort of words that make up the guide RNA itself. So we can look at A, C, G, and T individually, or we can start forming pairs or triplets, what you would call n-grams. And this is a fairly common approach, actually, in both malware static analysis, as well as in NLP and other text processing applications, which actually have really informed a lot of this work. They have a lot of sort of algorithmic similarities to each other. And one of the really cool things here is that we can go in and derive features from the sequence itself and sort of build up this representation of this molecule in a new vector space and then apply some of our machine learning approaches to it. So again, the other bit we have to take into account is that there are mutations. So we go and we want to make sure we take into account the roughly 4 to 5 million variants the particular cell line is. This has an influence on how we do our data weighting. There's also a lot of sequencing that needs to happen in order to pull that off. So you have these nice machines from Illumina that come in, they sequence the genome at various steps through the process to make sure that we're doing everything properly. These are the instruments that effectively do the reading of the DNA into a digital form, whereas CRISPR then is really about the writing. So in terms of infrastructure, we work on the Google Cloud platform, which has been really handy because we can use Google Cloud Storage to store these terabyte-sized genome files easily and move them around between instances. This is all orchestrated with Salt. We have sort of two teams at Desktop Genetics. So we have the tech R&D team, which is really the heavy Python team with bioinformaticians and data scientists who are prototyping these things out and doing sort of offline large batch computations with a pool of workers. And then you have the front-end team, which is really more JavaScript-oriented, and they're actually the ones who make Destron.com run. And so then we sort of use these cloud storage buckets as the interface by which we push data over from research into a production environment. Likewise, we would use the same system to push our trained models and things like that over. Within a particular worker instance, what you'll see is a collection of popular Python packages that some of you may also use. So the backbone of this is Tornado for the event loop. We also went and used quite a bit of C extensions, some of which are done in Python, some of which are just done in sort of regular C extensions to speed these things up because these genome files are quite big, and linked into a number of other tools to sort of make this all available over our API, and then that gets passed over to the front end, which does all the nice D3.js graphics and things like that. The other really interesting thing we found, though, is that when you're running a machine learning pipeline, there's actually sort of two systems operating in parallel. The first is the system shown here in blue. This is sort of representing our in silico model of the cell, and it's a very data-intensive system. It stores the genome, all of its annotations, features, and things like that. And it serves that data then sort of over to the actual machine learning pipeline, which is shown here in orange, that is really much more of a linear computational process that's going through a series of steps to go and extract features, build up the training data set, run through the regressions and things like that, and ultimately output something. And so it was quite interesting that you see that there's sort of these two orthogonal data science and data engineering sort of problems that need to be worked out and running in parallel. And that was one of the more interesting lessons that we learned. In terms of how we measure guide performance, we use an assay called the depletion assay. I'm going to skip over this, but we can come back to it if it's of interest. It's a technique that's been published a couple of years ago, where you effectively rely on an interesting property of evolution to go and measure how well a guide RNA performs. So if a CRISPR is really good at cutting the genome in a particular point, and you make a bunch of CRISPRs that cut genes that are essential to the cell to live, then if the CRISPR is good, then the cell dies. So then what you can do is make thousands and thousands of these guide RNAs in parallel and throw them at millions of cells. and measure the relative frequency of that CRISPR guide RNA in those cells over time. And if the CRISPR is really good, those cells will be depleted. They'll get taken out of the population, and you won't see them anymore. And that way you can come up with some nice numbers, and that's where our sort of output data comes from. It's quite noisy, though. We have to normalize for a number of factors. One of the really interesting bits here is that different scientists will take their time points at different days, and you have to find a way of coming up with a common statistic to normalize across that. You also have to take into account the fact that when a guide RNA is not very good at its job, it actually just sort of sits there and creates a lot of random noise, whereas if it was an active guide, you see a much stronger trend. And so there's a lot of signal processing effectively that has to go into sorting this data out so it can be cleaned up before it's actually fed into the machine learning pipeline proper. One of the other interesting bits we found is that whether or not a gene is essential for a cell to survive, and thus this whole test works, is actually a little ambiguous. We don't actually know all of the genes that are essential for a cell to survive, and actually they kind of change depending on circumstances. So what we had to do is find a way of finding the subset that seemed to have the most amount of evidence for it, and then we sub-selected those out. So we actually wound up using a very small percentage of the available data set to go and train this model, because it was the data set that gave us the most robust signal. And so our hope then is that we can generalize that result out to other CRISPR experiments. This is just an overview of the pipeline that I mentioned before. So we did a lot of normalization. We went through and filtered out a lot of things, and we had to come up with a way to reliably figure out whether or not a gene was essential and thus should be included in our training data set, regardless of whether or not it was terribly well studied, which is an interesting quirk. So what happens at the end of this is it actually worked amazingly well. We were quite surprised by this. So in this figure, just to break this down a bit, our approach is here on the left, and the current gold standard is shown here on the right. And what we're trying to do is predict the rank of guide RNAs in the human genome based on how likely they are to effectively achieve the goal of the experiment. And we did that by using the Spearman correlation coefficient as our test metric and plotting the actual performance versus the predicted performance versus the actual performance. And the way I like to explain this is that it's like we're running the Boston Marathon or the London Marathon, and we're saying, well, based on the prior rounds of this marathon, who is the most likely first place finisher, second place, third place, and so on. And what we find, though, is that even if you were to go and just have the same group of runners run several marathons, the fastest person doesn't always win. There is noise and fluctuation, trial to trial to trial, that has to be taken into account. And so what we had to do is try and predict something in that context. And so what we found is that we were able to get our correlation coefficient to around 0.7, whereas if you just did the sort of experimental data trials against each other, you're getting about 0.78. So we're now getting pretty close to the sort of limit and variability that we're seeing in the data itself, which is good. But it means that you still have to find a better or more precise way of labeling your data points, in a sense, if you wanted to push this even further. The other thing that was quite surprising is that the generalized linear models worked very well for this, probably due to the upstream feature engineering that we applied and data processing steps. And that convolutional neural networks or random forest models, which are more sophisticated and perhaps in vogue at the moment, didn't actually seem to show any significant improvement in this case. So simpler actually works very nicely if you do the upstream cleaning and processing of your data properly. We repeated this result as well for mouse. We actually got a bit better of a result there, but effectively it's the same finding. Now, another bit that was quite interesting is that when we looked at the actual weights that were coming up from our model, they seemed to be quite consistent with what we were seeing in the literature. This is good, and it's one of the nice benefits of these simpler models is that you can introspect them easily and correlate what you're seeing there to something that has, say, some biochemical meaning or that you've seen in other experiments and sort of know that you're on the right path, whereas some of the deep networks tend to be a bit harder to introspect and understand sort of what's going on inside the black box. One of the other interesting findings here is we did reproduce this effect, which is that the base pairs closer to the PAM site appeared to have a much more significant role on the performance of the CRISPR guide RNA itself, which is a good sign. That's what we want to see. One of the other interesting findings we found is that the performance of the model increased roughly linearly with the amount of data we were able to feed into the system, which is pretty consistent with a lot of other machine learning work. What was interesting is the human data set had a steeper slope than the mouse, so there was something about the way the model for the human genome worked that tended to be better, for lack of a better word, than the mouse. And we think this might be because just the human genome has had a lot more money put into it in terms of researching it and annotating it and curating it. And so really the quality of the data that you're working on seems to have a pretty significant effect here. The other thing that was, we did extend this further and eventually maximize them and didn't see really any significant returns beyond a point. And what we found then is that it's really also about the diversity of the data set that you're feeding into your model that had a really important impact and is something you need to consider. So just taking the same sequences and trying them over and over and over again isn't going to help. You actually have to try new experiments from other laboratories with other cells and things like that in order to sort of expose your model to the largest breadth of diversity possible. So in terms of some interesting findings and path forward, we found denoising and normalization was critical to the performance of the machine learning pipeline. The linear model performed surprisingly well, and it was coming in some cases close to the sort of noise you just see between different experiment trials. The model generalized well across different cell lines of the same species, so different humans, but it did not generalize between, say, human and mouse. They each had quite distinctive models, and we're not entirely sure why that is, because the biochemistry should be fairly similar. But nonetheless, that's a pretty interesting finding from a more scientific perspective. Prior knowledge about your genome of interest is important, and you sort of have this cyclical process where the more you know about your genome, the better you're able to edit it. The more you learn about the genome, that's the more you know, and so on and so forth. There's a sort of nice virtuous cycle here that we can really take advantage of. And that was really reflected then in our Python implementation, where you had to sort of have the sort of data engineering ETL pipelines running in parallel to your machine learning pipelines so they can sort of feed off of each other. And lastly, we saw the models increased with more data, which is good, but it was less so for mouse, and eventually it does limit itself, and it sounds like then you need to diversify the data set rather than just simply have more of the same. We also found some interesting Python lessons. The first was that things like Celery task queues, microservices, containers, and even the Postgres database actually really just increase the DevOps burden on the bioinformaticians to go and implement things rather than increase their productivity. Because as we've heard in some other talks today, when the code is changing so quickly and you're experimenting a lot, we actually found that just pure Python implementations without a lot of external dependencies or other services tended to be far more productive than something that might be more appropriate for a high traffic production case. And so the need to sort of simplify the infrastructure was actually far more important because it allowed us to move faster than adding sort of extra tools into our toolbox. The other thing we found, though, is that the portability of the scikit-learn models, which are serialized with cpickle by default, was not terribly portable because the ABI breaks between minor and patch versions. And this was a huge source of error in production. Not only are our models trained in scikit-learn, but many of the models coming from Microsoft Research and others in the field are, which is good. We're all using Python, but they are not very portable. And that makes it really difficult to reproduce someone else's results and sort of compare your approach to theirs and pass these things around. So we actually had to go and write some stuff to go in and extract our parameters into a more portable serialized format so that we can move these things around and have a robust and reproducible system. And we found that that was really an acute need here, was the ability to pull those parameters out and inspect them. We also found that Docker containers were not really a silver bullet when it came to replacing Python packaging. We had thought they might be, and we decided to give it a shot. Mostly just because for a lot of the bioinformaticians and data scientists, it puts a lot of DevOps burden on them, and it doesn't really solve the problem. You just sort of took one issue and you replaced it with a different one. And instead, actually, they found working with, say, like the Conda environment and the way the packaging was done there tended to be more effective. And then lastly, actually, the Google Cloud Storage or similar services like Amazon S3 were critical for this genome work. It's extremely I/O intensive. You have these huge data files that need to get moved around. And if you had to constantly be making logical volumes and things like that and doing hard drive maintenance just to support these terabyte data sets, it would be a huge pain. So being able to sort of stream these things on and off of these cloud storage buckets was wonderful. So going forward, CRISPR is moving forward into the clinic. There are a number of companies pulling this off into clinical trials later this year. There are a couple resources I recommend you check out, including the PySam library, which wraps HTSLib, which is the genomics library for all of this work, if you're interested in getting involved. There's also a number of places you can go to get access to actual genome data yourself to check it out. And with that, I would be happy to take any questions. Thank you.
And if you have any questions, come up to a microphone.
Now, you said your system was automatically taking into account SNPs and structural variations specific to that organism. Are you guys discerning those structural variants, large scale or complex, based off of haplotype of the sample, or is it just consensus?
So there are a couple ways we've approached it. In this specific project, the mission was to come up with something that would generalize across a number of cell lines that we were screening in. And so in that case, what we did is we go and take curated VCF files for those particular cell lines, and we merge those together to form a consensus. Now, handling very complex structural variants, like inversions and things like that, is a bit harder to do, because typically the way we would go about this is by patching the reference genome in to incorporate those variants at least a sequence level. And so when you have to, or we can also append alternative contigs and things like that to the file when we go and sort of build up our model. There are some really interesting projects around using more graph-like structures to represent the genome when you have complex structural variants, but it requires then rewriting very efficient string search algorithms as very computationally complicated graph search algorithms or subgraph search algorithms, and so there's sort of a big unmet need, shall we say, in that field.
All right, thank you.
I saw on the last slide you had something that said like, no lab, no problem. Can you
Yes.
talk about that a little bit?
Yeah, this has been a really interesting movement recently. So you know how the cloud has really revolutionized the ability to start a company in software because you don't have to invest millions in your own data center. Well, right now in the life sciences, you have to invest hundreds of millions of dollars in your own production facilities before you can even treat one patient. And so there are a number of companies who've been trying to provide lab infrastructure as a service. Transcriptic in the Bay Area is one. There are a couple others as well. And they actually expose this over a Python API that you can go in and remotely control lab robots, and then they bill you similar to Amazon or Google Cloud. And I think that's going to be one of the more exciting things because it would allow people to have access to this automation capability without having to go and raise, literally it would be about $40 million to sort of build out something like this.
Thanks.
So you might have touched on this in the first answer, but since there are individual mutations specific to every person, do you need to know these beforehand for every single person? Because I imagine the string matching has to be pretty precise, right? And these can be random mutations.
That's a good question, and I think it's still a bit open-ended right now as to, ideally, yes, you would. And there's different degrees of that too, because you can know the sort of genome of someone with different levels of confidence. So on one hand, you have, say, like the 23andMe assay that looks at a couple well-known mutations and things like that, versus, say, spending $5,000 on full 30x coverage whole genome sequencing in a CLIA-certified lab, or even going deeper than that. The idea, though, I think that's most important to get is that any information you have about the patient or the population genotype that you can incorporate has a benefit, and you should try to do so where possible.
Thank you.
Thank you.